Okay, well, welcome to this lecture. It's a real pleasure to talk about this subject. On the one hand, I'm a little bit nervous because there are various people in the panel, Shuba, Gemma and Tom in particular, but also Osvaldo and Lauren knows a bit as well, who are probably even better qualified to give this lecture than I am. But certainly primary production is a topic that I've become increasingly interested in over the years. And it's a real pleasure to talk about this during this seminar series, and in particular because Trevor Platt was a pioneer in quantifying and understanding primary production by phytoplankton in the sea at various scales in space and time. And so I just hope I can do some justice to the topic, given that this seminar series is part of the TPS Foundation. Okay, so the outline of this lecture, I'm going to keep it fairly simple because I'm a fairly simple guy, I guess. But I'm going to start by really talking first about phytoplankton biomass. Excuse me, I'm just going to shut my window because I've got a little bit of noise coming through from outside. There we go. Okay, so I'm going to talk about phytoplankton biomass and define what that is, how we measure that at sea and how we measure it from space, which will link back to a number of other modules in this training course. Before I then move on to primary production, and the reasons for this I'll go into when we talk about primary production, but it's often useful to look at primary production as a biomass normalized variable, particularly when we're modeling it from satellite observations. I'm then going to talk about the importance of primary production. So why is it so important and why are we doing this? Briefly touching on how we make measurements of primary production in the sea, and then from satellite observations, and then finishing by highlighting a number of resources that are available to the community, data sets that are available that you are free to play with afterwards. Okay, so like all my lectures at university, I like to sort of define some learning outcomes. So I hope that after this lecture, you have a good grasp of what primary production is, firstly, understanding the differences between phytoplankton biomass and primary production, understand how we measure it at sea and from space, and understand where you can get some satellite data from primary production should you be interested in pursuing this topic in the future. Okay, so starting with the definition of phytoplankton biomass. So the biomass of phytoplankton can be described as its sort of local abundance. The equivalent term in fisheries is often used as stock size. So it's sort of a snapshot in time of the amount of phytoplankton in the water. Now, many metrics exist for phytoplankton biomass. Two commonly used metrics are carbon, but also nitrogen as well, because a lot of modelers like to work in flows of nitrogen in their ecosystem models. Carbon is a useful metric because it's an abundant element. If we look at the Redfield ratio, for instance, there's a lot more carbon than there is nitrogen or phosphate. However, there are some disadvantages with the metric of carbon in that it can be quite tricky to measure directly in the ocean. Now, in terms of phytoplankton biomass, it's interesting often to compare the stock of biomass or the amount of biomass of phytoplankton in the ocean with the amount of biomass of terrestrial plants on land. And if we do that, we can see that actually the biomass of phytoplankton in a snapshot of time in the ocean is minuscule almost compared to the amount of biomass of plants on land, terrestrial plants here. But that, as I'll allude to a little bit later on, doesn't in any way mean that phytoplankton are not important for primary production. Now, in terms of a common index used for phytoplankton biomass, and it's an index that I'll continue to use throughout this lecture, is that of the chlorophyll A concentration. The concentration of chlorophyll A pigments, so photosynthetic pigments in phytoplankton, is often used as an index of phytoplankton biomass. The reason being, well, a number of reasons. Firstly, it can be associated directly to phytoplankton in the ocean, unlike carbon, which is in bacteria, it's in zooplankton. And so it's a fairly easy metric to measure in the ocean. And also it has a direct link to ocean color and consequently how we can observe phytoplankton biomass from space. Okay, so how do we measure chlorophyll A concentration in the sea? Well, there are a number of ways of doing this. Often it requires going out into the ocean and collecting water. Now that can either be from a CTD rosette, so you lower the CTD vertically throughout the water and you snap the bottles of the Niskin bottles shut at various depths, and you can take that water on board and you can filter it, and then you can put the filters into some chemicals to extract the chlorophyll concentration, and then you can measure that in a pherometer. That's one way of doing it. Another way of measuring chlorophyll using fluorometry is to do it in vivo. So to measure it directly, either using a fluorometer attached to the CTD or potentially on a underway, non-toxic underway sample. So if you look at this image on the top left here, this is just sort of illustrating how water can be pumped through a ship as it moves, and you can continuously measure chlorophyll fluorescence. Now, there are advantages and disadvantages to both these techniques. Typically, the in vitro method, the extraction method, is a lot more accurate for measuring chlorophyll in the ocean and phytoplankton biomass. Another way of doing it, which is commonly adopted in satellite validation protocols, is to use high performance liquid chromatography. So similar to the fluorometric methods, you filter onto a GFF or glass fiber filter. You typically flash freeze it in liquid nitrogen, put it in the -80 freezer, and then when you have the time, you can take that sample out, you can put it in some chemicals to extract the pigments, and then use column chromatography in order to separate out those pigments and measure them using high performance liquid chromatography. This way you can also look at other pigments in the phytoplankton sample, which is useful for looking at the phytoplankton composition or looking at the physiological state of the phytoplankton in the water. But this is a common technique that's used to measure chlorophyll concentration, the metric of phytoplankton biomass that we're going to be using in this lecture. Now, more recently, or in more recent years, another technique is increasingly being adopted, and that is in vivo spectrophotometry. So here you use a spectrophotometer sort of in line. So this is an example, I think, on the bottom left of an ACS, where water is can be pumped through the ACS, and it measures the absorption coefficient of particles if you do filter no filter system, and you can get very accurate measurements of particulate absorption as a function of wavelength, shown on the top right there. And of course you have this absorption bump in the red, which is directly related to the chlorophyll A pigment. And if you look at the height of this bump, it's very tightly correlated with the chlorophyll concentration. So more recently, people have been attaching these types of instruments onto the CTD rosette to measure profiles of chlorophyll in the ocean, but also attaching them to the non-toxic underway water supply to get very accurate continuous measurements of chlorophyll A phytoplankton biomass as the ship moves through the ocean. Okay, So that's a little bit about how typically we measure phytoplankton chlorophyll A concentration at sea. Now, in terms of how we measure the chlorophyll concentration from space, I know some of this will touch on previous training modules, like Module 2 here, which gave us an introduction to ocean colour. But how we do it from satellite is simply by looking at the reflected light from the ocean surface and then relating that to the chlorophyll A pigment. Now, chlorophyll A absorbs very strongly in the blue and a little bit in the red. So what generally happens is the more chlorophyll concentration in the water, the more blue light gets absorbed, and hence that impacts the reflected light that the satellite sees, and the oceans begin to look greener. So this is an example of an image actually took from my phone in the center of the North Atlantic gyre. And as you can see in this image, the waters look very blue, and this is because there is not very much phytoplankton biomass in the water. Chlorophyll concentrations are very low, so the optical properties of pure water dominate, and the reflected light seen from a phone or seen from a satellite looks very blue. As the chlorophyll concentrations increase, this is another photo taken in very high chlorophyll waters. We can see that a lot of that blue light gets absorbed, and the oceans begin to look green. And so by looking at spectral variations in the remote sensing reflectance measured from the satellite, we can get pretty good approximations of the chlorophyll concentration. And that's just shown again in an example here. So this is from one of Schuber's papers published back in 2017. It's a very large in situ database of the chlorophyll concentration shown on the y-axis here, plotted as a function of essentially the ratio of blue to green lights. This is the maximum band ratio. So basically what's plotted on the x-axis here is a ratio of blue to green light, but it's switching between the blue wavelengths 443 to 490 to 510, depending on which wavelength has the highest signal. And so what you can see in this image is that the chlorophyll concentration is quite well correlated with the ratio of blue to green light. Now, there's a lot of scatter around these relationships, which are down to a number of factors, some of which you possibly touched on, maybe when you were looking at the module or listening to the module on optically complex waters, where we know that this ratio of blue to green light is also sensitive to other constituents in the water. So if there's a lot of CDOM in the water, for the same chlorophyll concentration, it's going to modify that ratio. If there's low amounts of CDOM, it's going to affect it. If there's high amounts of non-algal particles, and so on and so forth. So it's a model that's very much based on what they used to call a case one assumption, where phytoplankton are the principal component in the ocean controlling the optical properties. So some of the first satellite observations of chlorophyll A concentration came with the launch of the Coastal Zone Color Scanner by NASA in the 1970s. And sort of illustrated in these two publications by Hovis and Gordon in Science in 1980. And this was really revolutionary at the time. First examples of how we can, or among the first examples of how we can use satellite observations to monitor the biomass of phytoplankton over large scales. So you can see here where Florida is just in the image here. And on the right-hand side, the image was matched alongside a transect of chlorophyll measurements collected by a ship using photometric-based measurements. And you can see how well correlated the spatial changes in chlorophyll are when matched between the satellite and the ship. Okay, in more recent years, we've now are blessed with a suite of very highly accurate and well calibrated satellite ocean color missions. These are just some examples of Sentinel-3 in the top left, MODIS Aqua and VIIRS. And as you can see in this nice image in the middle, how the spectral variations or the color observed by these satellites can be linked to the concentration or the biomass of phytoplankton in the water in the context of the chlorophyll concentration. Those very green waters being seeing the presence of much higher phytoplankton abundance, a lot more chlorophyll in the water than the blue regions. And I pinched this slide from Tom Jackson's talk last week, but now we're blessed with nearly 25 years of continuous ocean color observations, and with projects like the Ocean Color Climate Change Initiative, where they've strived to successfully merge many different satellite ocean color missions together to produce this continuous time series of chlorophyll concentrations. We can really get detailed patterns of how the phytoplankton biomass in the ocean changes seasonally, changes spatially, and also changes at interannual timescales. And that's sort of illustrated in this figure here. So this is a figure taken from a paper published back in 2016. And we're just focusing on two different regions of the ocean here, the equatorial Pacific on the top part of the image, and also the Indian Ocean on the bottom. And what we've plotted here are climate indices. On the top right is the multivariate El Nino La Nina Southern Oscillation Index as a function of time over the periods of 1980 to 2020. And on the bottom is the Indian Ocean dipole, which is essentially a difference in sea surface temperature between the two boxes shown in the bottom left. And what we've done is we've overlaid the time series of chlorophyll concentrations. This is again from the Ocean Color Climate Change Initiative. This was an earlier version of the data set. And what you can see is that interannual variations in climate are very tightly coupled to the chlorophyll concentration, the biomass of phytoplankton in the water. And one of the reasons this is because the lifespans of phytoplankton are relatively short compared to plants on land, and so they are capable of responding very rapidly to variations in climate. Okay, so that was a bit about phytoplankton biomass. So getting kind of into the meat now of the lecture, I'm going to begin talking a bit about primary production now. What is primary production? Well, primary production is basically the rate of photosynthesis of phytoplankton. It's the equivalent term in the terms of fisheries is sort of the reproduction and growth of the fish. I like this term. I think that was coined by Schubert and Trevor a while back, saying that if biomass is like money in the bank, primary production is like the interest earned by the capital. So it's a sort of nice way of separating out what biomass and primary production is. Now, one of the very interesting things with phytoplankton, if you recall earlier on in this lecture series, we talked about how the biomass of phytoplankton is minuscule compared to the biomass of all plants on land. Well, when it comes to primary production, things are very different. Primary production by phytoplankton is pretty much equivalent in the ocean to that by terrestrial plants on land. So both of which contribute around 50 gigatons of carbon to global primary production. Now, the reason this is, again, comes back to a similar point I made in an earlier slide, that phytoplankton have very short growth cycles. They're constantly growing and dying, whereas the life cycles of terrestrial plants are much longer. And as a result, their primary production of phytoplankton is equivalent, or thought to be equivalent, to that of terrestrial plants on land. So they are a major player in a global planetary primary production, as illustrated by some of these references shown on the bottom there. So what is primary production? Well, we talked about the rate of photosynthesis. So what is that? Well, it's basically the conversion of inorganic carbon from carbon dioxide into organic carbon with light as the energy source. So in the ocean, this is typically expressed in terms of mass of carbon converted or fixed per unit volume of water per unit time. So it's a rate. And if we look at the basic photosynthesis equation and how it works, we can see that we have water, H2O, and we have CO2. We use the available energy in the form of sunlight that's absorbed by the phytoplankton through the process of photosynthesis. And these elements and compounds are converted into carbohydrates, stored energy, plus oxygen. So oxygen is produced and dissipated energy. So that's the sort of basic equation of photosynthesis. So like plants on land, the growth requirements of phytoplankton are similar. They require water, which of course is extremely abundant in the ocean, not a problem. They also require carbon dioxide. Again, the carbon dioxide concentrations in the ocean are quite high, and so that's available. They need light. Now that potentially becomes a bit of a problem because light gets attenuated quite quickly in the ocean. So phytoplankton are only able to grow where there is available light at the surface of the ocean. And like the plants on land, they require nutrients, not only macronutrients like nitrogen and phosphorus, but they also require micronutrients. So essential nutrients like iron, even though they only require low concentrations of iron, they're very important in the process of photosynthesis. Okay, so a few definitions before we move on of primary production, because there are various aspects of primary production in the ocean. The first one I'm going to talk about is gross primary production. So this is often defined as the total rate of organic carbon produced by autotrophs or by phytoplankton. So it's the total amount of primary production. Then there's net primary production. So this is referred to as the total rate of organic carbon production by phytoplankton, by autotrophs, but minus the respiration by phytoplankton. So respiration is, of course, the inverse process of photosynthesis, whereby oxygen is converted back into CO2. So with net primary production, you subtract the respiration away from the gross primary production in order to get net primary production, but the respiration only by phytoplankton. Now, another term commonly used is that of net community production, and that is defined as the total rate of organic carbon production by autotrophs by phytoplankton minus the respiration by not only phytoplankton, but all the organisms in the ecosystem. And another term we often refer to is export production, which is defined as the amount of organic matter produced by primary production in the surface ocean that is lost to the deep ocean. So I thought it's important just to make some definitions of those terms before we move on. I think I saw a question pop up about that as well, so it was quite good timing. Okay, so what is this process of primary production then, and why is it important? Well, this is a sort of schematic diagram. I think Tom might have shown this one last week as well, of the ocean carbon cycle. a simplification of the ocean carbon cycle. And where does primary production lie? It's lying right about here in this process. So we have our dissolved inorganic carbon, which is being converted into particular organic carbon through the process of photosynthesis. So this is kind of like our gross primary production. And then you would minus the respiration just by phytoplankton here in order to get your net primary production. Now, this little process in the middle arguably is driving a lot of this much larger ocean carbon cycle. So the production produced by phytoplankton then gets consumed by zooplankton. That can then you then have a whole suite of quite complex processes where bacteria are breaking down some of that carbon. There's a lot of respiration going back in the form of DIC. There are DOCs being produced through bacterial remineralization. And some of this material is then sinking out of the surface layer, forming our export production, either directly by the phytoplankton or aggregating in the form of fecal pellets, for instance, by zooplankton, pooing out a lot of the material that's been eaten. There's also complex vertical migration going on with the zooplankton, where they come up at the surface during the night to feed and then go down to depth and excrete some of that carbon that's been eaten. There's also physical processes moving around these different pools of carbon in the ocean, and there's a whole lot of complex processes going on at depth as well, resulting eventually in an element of that primary production produced in the surface being transferred into the deep ocean in the form of export production. Complex processes going on, but essentially the engine behind the whole of this is the primary production by phytoplankton in the ocean. The part of the image on the right here is just sort of showing various ways we can monitor these pools and fluxes of carbon in the ocean, satellites being a big focus of this talk here. Okay, so why is this primary production important? Well, I sort of just touched on that then, but I'm going to go in a little bit further detail here. There are many reasons why primary production is important, but I'm just going to focus on two key reasons here. Firstly, I'm going to talk about the ocean carbon cycle. So what happens? What happens with phytoplankton in the surface layer? Well, phytoplankton are photosynthesizing. So that's drawing CO2 out of the water, and it's modifying the gradient in CO2 in the ocean. So this is an important point to make first, because this disequilibrium caused by production of phytoplankton at the surface reduces the CO2 concentrations at the surface, which can, of course, promote air-sea gas exchange. Now that primary production produced by phytoplankton at the surface, as we showed in the previous slide, an element of a large portion of that will get respired back from organic carbon into inorganic carbon, so going back to CO2, but a certain amount of it gets exported down into the interior ocean. So it's thought around 50 gigatons of carbon is produced in the surface of the ocean. Around 10% of that gets exported into the interior ocean. And this continues to sink and gets quite heavily remineralized, so converted back into CO2 as it sinks down towards the sea floor. And only about 2 gigatons is thought to be deposited in at the surface, at the bottom of the ocean, and around only 0.2 getting buried into the sediments. But one of the important points to note here is that the further this carbon gets down into the deep ocean, the likelihood is that it will be kept away from re-emission into the atmosphere for a longer period of time. So basically, there's increasing timescales of ocean carbon storage as that particular organic carbon sinks down to the seabed. So This process of primary production and the ocean biological carbon pump is very important in controlling the surface CO2 concentrations and consequently the ability of the ocean to absorb CO2 from the atmosphere. So that's one important reason. Now, another important reason is fisheries, primary production are at the very base of the marine food web, right? So all higher trophic levels are dependent on their productivity. And there's this classical sort of 10% law. They say that 90% of the energy gets dissipated as you go from your primary to your secondary to your tertiary trophic levels. But ultimately, the production by phytoplankton at the very base is critically important for feeding, for sustaining the entire marine ecosystem. And this is all the way through to humans through the seafood we eat. So this is a nice paper I came across, I think last year, and it was some work conducted in the US where they showed how the average annual productivity here, plotted on the x-axis, is well correlated with the fisheries revenue in the US. So the higher the productivity, the larger the fisheries revenue. I mean, of course, there's many other factors that impact fisheries, but it's just to illustrate the importance of primary production in this process. Okay, so on to measuring primary production at sea. Well, one of the first kind of ways people did this, at least from reading a little bit of literature, particularly from this very nice book by Mills in 2012. If you're interested in learning a bit more about the history of biological oceanography, I strongly suggest you read this book here by Eric Mills. It gives a wonderful history of the early biological oceanography. But one of the ways people got an index of phytoplankton productivity originally was to count phytoplankton cells and then make some sort of assumptions about the productivity per cell in order to get an idea of primary production in the ocean. But one of the interesting methods developed, I think, back in the 1930s was by H. Gran, a Norwegian scientist, I believe, who made use of the fact that oxygen varies with the abundance of phytoplankton, of course, oxygen being a product of the process of photosynthesis, and so can be used to get quite a quick bulk index of primary production. And so he developed this light and dark bottle method, whereby you would sort of suspend bottles at various depths. Often the bottle right at the bottom you would cover up so you can get some idea of respiration, and then you would monitor each bottle with known concentrations of phytoplankton cells, known salinity and oxygen content, and then you would look at how that would change over a period of time in order to get a proxy of primary production. So this was one of the earlier techniques known as the light and dark bottle method. Now, I wanted to link back to the module 7 by Grints and George on water quality and human health. He talked a bit about Secchi discs in that module as well. So some of the earlier work in the sort of 80s and 90s also, and this was some of the first approaches that looked at how clarity of the water in the ocean can be linked to productivity. Given that there was this long history of Secchi depth observations going back to the late 19th century, there was various work that would relate the Secchi depth to the phytoplankton biomass and then make assumptions about the productivity per unit biomass in order to get an idea of how phytoplankton productivity was changing in the ocean. The paper I just pick out here by Fakowski and Wilson in 1992, which was sort of a response to an earlier paper suggesting that the phytoplankton productivity was increasing in the North Pacific, and they studied the Secchi depths from a long time series and found that actually they weren't seeing any significant changes in Secchi depth, implying that the productivity in the North Pacific wasn't changing drastically. So a link there to some of the earlier work that was taught in this training module. Now, in terms of measuring primary production at sea, in the sort of modern era, there are a whole range of techniques to do it. And I don't really have the time to go into all these different methods. The 14C method, I'll talk about it maybe briefly in a little bit. But this is a table that was taken from a recent paper published by Schuber, a book chapter published by led by Schuber in 2019 that lists the various methods we can measure primary production in the ocean. Again, with reference to the different types of primary production: gross, net, and community production, and export production, and on the various timescales that these measurements need to be conducted at in order to get at the production property. In the same book, actually, in the same book, Encyclopedia of Ocean Sciences, there's also another chapter by Church, Cullen and Carl, which also talk about some of these other techniques as well, optical techniques, as well as the sort of standard 14C methods and so on and so forth. So if you're interested in the subject and want to learn a bit more about how we measure this in the sea directly, then I really strongly suggest you have a little look at these two book chapters. The other thing as well is that the IOCCG have just published a protocol series on measuring primary production at sea, volume 7, Aquatic Primary Production Field Protocols. This is really an excellent resource. I strongly recommend you looking at, and they have this nice figure in one of the earlier chapters which talks about each of these different methods of measuring primary production in the ocean and how they vary on temporal and spatial scales. Now, in terms of my experience, I haven't had a lot of experience in measuring primary production directly at sea. I'm normally one of these individuals who likes to play with satellite images instead, but I have been on a few of these Atlantic Meridional Transect cruises, and these are just some of the ways we did it on those cruises on AMT. So this is a figure I actually took from Gavin Tilstone, who would come on board the AMT and do a lot of these on-deck incubations. So what you would do here is you would collect a water sample pre-dawn, before the light, before the sun rises, and then you would incubate them on deck over the course of the day. So you would use the 14C technique, whereby you add 14C to the sample, and basically you run these incubations over the course of about 12 to 24 hours at various light gradients that are designed to reflect the depths at which they were collected in the water. And then you would look at how much of that 14C was taken up in the form of particulate carbon by measuring it on a scintillation counter afterwards, from which you can get approximation of the primary production. Another common technique that's used are much shorter term photosynthesis irradiance experiments run in photosynthotrons, whereby you take your sample and then you incubate them over a light gradient, and you do that normally on a much shorter time period of 1 to 2 hours, and then you, again using the sort of C14 method, you measure the primary production, and then what you typically do is you fit what they call this photosynthesis irradiance curve, which I'm going to start talking about in a minute, whereby the irradiance is plotted on the x-axis as a function of the production on the y-axis. This is production normalized to biomass. So, and then when you plot this curve, you fit what they call a photosynthesis irradiance curve, which again links back to the second slide I showed. Trevor Platt was a real pioneer in how we fit these functions to the data and how we can interpret the parameters of the function. So here we have the assimilation number and the initial slope, and IK, which I'll talk about in a moment. Now, one of the slight issues with all these primary production measurements at the sea is often they're measuring slightly different aspects of primary production. The incubations are at different periods of time, so it's very hard to reconcile all of the different techniques for measuring. But I just refer you to a nice paper by Jarko in 2016. So there are some nice work that's being done where they use mathematical modeling to start to more directly relate these different types of primary production measurements. Okay, so that was a bit about measuring primary production in the sea. Now I'm going to talk about how we do it from satellite. So I first wanted to show one of the first maps of global primary production from space. So this was published by Alan Longhurst, Schubert and Trevor and colleagues back in the mid-1990s. And it was really quite a revolutionary study. There were subsequent studies published in 1996 and 1997 by David Antoine and Mike Berenfeld and Furkowski. But these were the first sort of real illustrations of the power of satellite remote sensing, the ability to use satellite ocean color observations to kind of for the first time get very, remarkably accurate estimates of global primary productivity. Using satellite remote sensing directly, but also as an extrapolation tool for our in situ observations. And at the time, they quantified global primary productivity to be around 50 gigatons, 40 to 50 gigatons of carbon per year, which, 25, nearly 30 years later is a number that we're still arriving at with our satellite observations. Okay, so now there are many ways or many different types of models for primary production from space. And I'm going to stick with a very simple one for the last part of this lecture here. But I would just like to, I'll refer you, if you're interested in learning a bit more about these different approaches, to a paper by Schubert and Trevor published in 2007 in Oceanology, which really was a nice piece of work, in my opinion, because what it showed is that although there are very different techniques used to measure satellite primary production, essentially they all boil down to the same 3 or 4 variables / parameters, and they are fundamentally similar. So I'm going to use an available light model for the rest of this lecture, which is quite a fairly simple approach, but it incorporates all of the key aspects of modeling primary production from space. And at this point as well, I hope these next few slides illustrate why I separated out phytoplankton biomass from primary production in the earlier part of this lecture. So it's common in computing primary production to work with biomass normalized production, so what they call PB here. And this is just to sort of illustrate that we do that by dividing the primary production by the biomass to give ourselves PB, and we can reconstruct the primary production by knowing the normalized primary production and the biomass. And one of the key reasons we do this is because we can get good, accurate estimates of chlorophyll concentration from space. So we can use our biomass as a scaling factor if we know the normalized primary production, biomass normalized primary production. Okay, so I'm going to now work with a little bit with PB rather than with P. So production normalized by biomass. And as I mentioned in a couple of slides earlier, this is what they call a photosynthesis irradiance curve. Again, a lot of work by Trevor Platt and colleagues in the 80s and 70s, looking at different ways of quantifying this. But here we have normalized production on the y-axis as a function of irradiance on the x-axis. And if we plot our primary production measurements, we can fit what they call a photosynthesis irradiance curve. And this has 2 essential parameters. The initial slope, which is often linked to the efficiency of the phytoplankton in primary production, and the assimilation number, or known as PB max, which is often referred to as the capacity of phytoplankton for primary productivity. And we also have this parameter here, IK, which is essentially a ratio of these two parameters and gives us an index of the light levels at which the phytoplankton are acclimated at. Now, this is the mathematical formulation of that curve I showed you in the previous slide, this green curve here, where our normalized production PB is modeled using this exponential equation, PB max here, that assimilation number, and alpha B here, and then our irradiance up here on the equation. So assimilation number of the light saturation curve, PB max, the initial slope, and the level of light. So going back again, light I, PB max, maximum, and then this initial slope alpha B. Okay, so one thing to bear in mind is that many of these parameters and variables vary with time and with depth. To keep things simple in this example, our PB max is varying with depth here, and so is our alpha B, and our light irradiance is varying with depth and time here. Now, of course, we could look at spectral variations in light. So if we had a spectral primary production model, these two parameters would also be wavelength dependent, but we're keeping things simple here. So we're dealing with light in the form of PAR, photosynthetically available radiation, so light integrated over the visible part of the spectrum. Okay, so if we want to know the primary production in units of meters squared per day, we have our same equation here, the photosynthesis irradiance curve, PB max and alpha B and light. And now, of course, we're converting back to our primary production here. In order to do that, we need to scale to our biomass, right? Because this is the normalized primary production. Our biomass also is going to vary with depth. And then we integrate over the day length D, and here I'm using the euphotic depth, but you could integrate further down into the water column, but just bear in mind that as you get below the euphotic depth, there's not much light for photosynthesis anyway. So just for the purpose of this simple example, we're going to integrate down to the euphotic depth. Do bear in mind that this simplification does not account directly for spectral variations in light-driven photosynthesis, as I just mentioned before, and assumes the photosynthetic parameters and the biomass do not change over the course of the day, when, of course, in reality they can. Okay, so how do we do this then from satellite? Well, first of all, we need to know the day length, right, as part in the first integral here. So we can get a good proxy of day length simply as a function of latitude and time of a year. So we can model that pretty well. The next thing we need to know is the depth of the euphotic zone, the 1% light level. And again, this can be estimated pretty well from satellite ocean color data, actually. For example, using estimates of the diffuse attenuation coefficient, and the euphotic depth is now a sort of standard product actually produced by many space agencies. So day length and euphotic depth we can get quite well from satellite. Okay, so our next parameter we're looking at is PB max. Now this is a bit of a tricky one. It's not so easy to derive directly from satellite ocean color data, and as a result, there are many different ways at which we've tried to produce maps of PB max or the assimilation number using different techniques, which I'm going to go through in a minute. The first technique I'm going to show here is using other environmental data. So there's work that has looked at the relationship between PB max and sea surface temperature and irradiance. This is built on sort of some of the early work initially by Epley and others. We can get very accurate estimates of sea surface temperature and PAR from space. And so if we know that from satellite, we can use these models to sort of predict what PB max is from satellite and then feed them into our primary production model. Another approach, actually, this was a nice paper published by Trevor back in and colleagues in 2008, is to use large in situ data sets of PB max and then sort of develop techniques that look into this data set and find, for a given pixel, similar conditions. So if we can get chlorophyll and temperature in a pixel accurately from satellite, we can then scrutinize that in situ data set to find conditions with similar chlorophyll and similar temperatures, and then find or search for an adequate PB max, a measurement that was conducted under those conditions, and assign that to a satellite pixel. Now, another technique is thinking about phytoplankton community structure. Now, next week, I believe Heather Bowman will be talking about that in module 11, about phytoplankton community structure. But what we typically see is that different communities of phytoplankton in the ocean photosynthesize it in different rates. And so the parameters, both the initial slope and PB max, vary depending on the size, in this case, in these examples shown here, but also the taxonomic composition of phytoplankton in the ocean. So if we know that, if we can predict what the type of phytoplankton is in the water from space, we can really improve our assimilation number estimates. Now, one of the another approach that has had a lot of success and sort of goes back to that map I showed you by Alan Longhurst, the first global satellite estimates, is to use biogeographic provinces or biogeochemical provinces. And so these are regions of the ocean which are relatively homogeneous. And so if we have data collected in that specific region, we can use these, we can average that within the region and feed that into our satellite primary production models. Not only regionally, but also seasonally as well. So we can look within a given province at how the PB max varies over the course of a season and feed that into our satellite primary production estimates. Similar to PB max, alpha B is another parameter of this satellite photosynthesis primary production equation that's a little bit tricky to get at. Though there are some nice recent work showing how well actually estimates of light can help. But these, again, just three ways at which we can estimate alpha B. So this initial slope, the efficiency of the phytoplankton, we can estimate that parameter and feed it into our primary production estimates. Again, using the nearest neighbor technique I mentioned before, or looking at phytoplankton community structure, or thinking about aggregating the in situ data sets into different biogeochemical provinces. Now, there is a lot of exciting work actually going on, and I know there's going to be a module 16, I think, looking at artificial intelligence, but a lot of promising work showing how well AI-based or machine-based learning may be able to help in our estimation of these two photosynthesis parameters, alpha B and PB max. Okay, so let's say we solve that. We now need to know the light environment. Now, fortunately, we can get very good, accurate estimates of photosynthetically available radiation from satellite data. And so we can quantify the light environment just above the sea surface. Now, if we know that and we can make good approximations of the diffuse attenuation coefficient, another key variable that we can get from ocean color satellite data, we could begin to propagate that surface light down through the different depths in order to model our light environment accurately, driven by satellite observations. And of course, this diffuse attenuation coefficient may vary with depth as well, right? So in some regions of the oceans, we have a deep chlorophyll maximum, like I'll show you in a minute. So we even have models that can consider depth variations in diffuse attenuation coefficient in order to improve our estimation of the light environment in these models. And finally, our biomass. I talked about a bit earlier about this importance of scaling. and to phytoplankton biomass. So we can get chlorophyll concentrations very nicely at the surface from space. But of course, that's only at the surface. If we want to know what's happening further down the water column where the satellite can't see, but whether there's still light available for photosynthesis, we need to be able to model or to extrapolate those surface observations down to depth. And there are various empirical, semi-empirical, and machine learning based methods that are now being introduced, driven by not only satellite observations, but also in the increasing use of ocean robotic platforms like biogeochemical Argo floats in order to help that extrapolation of the surface fields down through depth. Okay, so if we have all that information, then on a per pixel basis, we can begin to quantify a global primary production. This is just an example of an estimate of primary production from May 2004. So what we can see here is we're going into the spring bloom period in the North Atlantic, and we see very much higher rates of primary production in the North Atlantic, where the day length has got quite long and where we have the phytoplankton spring bloom. Of course, we can use satellite observations like we showed earlier in Tom's nice video of chlorophyll concentrations. We can do similar things for primary production. So we can begin to look at the seasonal variations in primary production. This is an example from a model where we estimate the primary production from large phytoplankton. And we can sort of see quite clearly in this image in the Atlantic Ocean how the seasonal variations of phytoplankton primary production varies over the seasonal scale. Okay, so in terms of validation, one of the critical things with all satellite products is we need to be able to have good in situ data sets on primary production to evaluate and validate our satellite observations. There was a lot of effort by NASA in the sort of mid-90s through to sort of, I think, 2011, where they ran a series of primary production round robins. They tested different primary production models and validated them. And it's commonly assumed nowadays that the uncertainty in primary production is around 30% in log 10 space, around 50% in linear space. Though I have to say there's a lot of work going on now improving our estimation of uncertainty in primary production. And I know Gemma, who's on the panel, has been doing a lot of nice work on that recently. So this is very much a moving field. And this is just an example of sort of our most recent estimates of global primary production from a very nice paper by Gemma in 2021 using the Ocean Color Climate Change Initiative data. And they quantified global primary production to be in the region of around 48 to 53 gigatons of carbon per year. Again, very similar to that estimate of Longhurst back in 1995. Okay, and so one other thing to mention, we talk mainly about primary production, but there's also increasing work looking at how we estimate that export production, the amount of production that's in the surface layer that gets exported below the euphotic zone. And there are techniques that people have developed to do this. This EF here is what they call the export production to primary production ratio. And the people have developed empirical approaches to estimate this, driven by satellite temperature and primary production estimates. So you can convert your satellite primary production estimate into an estimate of export production. And a lot of work going on this topic in the European Space Agency BICEP project. Recently, they've been testing an empirical approach by Dunn et al. In 2005. And that brings me nicely on to the last point of the lecture, which is just to illustrate some of the resources and primary production that are available to the community. These are 3 data sets that have recently been produced in the BICEP project. There's a monthly global primary production data set based on the work by Gemma in her 2021 paper. Monthly primary production from 1998 to 2020 at 9 kilometer resolution, all freely available. Additionally, we've just also in the project published a monthly global export production product as well. Again, going from 1998 to, but this time to 2019. And there's also products on phytoplankton biomass, not only in terms of chlorophyll concentration, which we can get from the Ocean Colour Climate Change Initiative, but also within BICEP, the phytoplankton carbon concentration, so estimates of phytoplankton carbon. Also, some other resources here. NASA run a site on ocean productivity, which has some other primary production products. Again, really encouraging you to go and look at this in situ aquatic primary production field protocol series. It's really excellent. And there's a link directly down there on the resources button. And also just to let you know that we've recently had an IOCCG working group approved in March 2023 on primary production. And so we hope to be producing one of these nice IOCCG reports in the coming years on primary production. So that's it for the lecture. Just a very brief overview. We've talked about our definition of phytoplankton biomass, how we measure that in the sea, dealing mainly with chlorophyll concentration in this lecture, how we measure that fluorometrically using high-performance liquid chromatography or in vivo spectrophotometry, how we measure phytoplankton biomass from space, linking back to some of those early modules that you would have learned on relating the colour of the ocean to the chlorophyll concentration. Definition of primary production, this conversion from inorganic carbon to organic carbon through the process of photosynthesis, why it's important for the carbon cycle for fisheries, how we measure that in the sea, going back to some of the very early techniques of the light and dark bottle method, all the way through to the various methods they use today. Again, didn't go into that in too much detail, but the resources are here available in these slides. And then a bit on how we measure it from space using a simple available light model and some resources. So at that point, I'll stop. I think I've been just over the hour, so I think it's a nice point to stop there.